Welcome to STEMpaths, the show where we have students unpacking papers, study paths, and the people behind them. I am Dr. Thornton Larson, and with us today we got our guest hosts, Ruby Dominguez and Hayley Hawkins, and today they are going to be interviewing Ian Gazeley, who wrote his paper, Conservation Genetics of Roosevelt Elk Population and Isolation and Reduced Population Isolation and Reduced Diversity. Man, I can read and speak correctly. So anyway, ladies, if you'd take it away.
Okay. Yes. Thank you, Professor Larson, for the introduction. My name is Hayley Hawkins, and I am a senior majoring in biology. And this is my partner,
Ruby Dominguez. I am also a senior and also majoring in biology.
Yes. And before we get to the summarization of the article, I just want to say thank you so much for taking the time to be here. You could be doing anything else, and you're here with us, so greatly appreciate it. From what I gathered, a study on Roosevelt elk conservation genetics in British Columbia found that reintroductions led to notably lower genetic diversity and heightened genetic drift, especially between the source population on Vancouver Island and the reintroduced mainland groups. The analysis showed genetic isolation of the southern Vancouver Island herd and significant genetic differences among reintroduced groups, suggesting that the reintroduction approach has produced isolated populations with reduced genetic variation, making them more susceptible to inbreeding and associated negative fitness effects.
And I also want to add to that as well. One of the most striking findings in the study is that Roosevelt elk populations in the Vancouver Islands still retain the majority of the remaining genetic diversity for the species in British Columbia, despite their own historical decline. This makes the island populations extremely important for conservation because they act as a genetic reservoir, a source of diverse genes that can help improve the health of more inbreed populations in the mainland. Finding these highlights in a critical point of wildlife management. Conservation isn't just about the numbers, but it's about the genetic diversity.
Okay, so do you feel like that covers all of the necessary points, Mr. Gazeley?
I think those are both good summaries of kind of the main point of the paper, of the research. There's a lot of detail that is involved. But yeah, to summarize it down to a few words, very good. Very good. I think
okay.
Were those elements intentional focal points?
Sorry, what was that?
Were those elements intentional focal points, the ones that we mentioned in our summary?
The intentional focal points of my work?
Yes.
Yeah. Basically that despite the grand success that the reintroduction reintroduction project and the recovery project is, and it's continued to be extremely successful, there's concerns. So what I was looking at within that paper, the real focus was like, what are the genetic consequences of the approach that was taken to reintroduce those animals? So.
Now, I did have my interesting fact would have been the technology behind it. From what I understood, you guys used microsatellite markers.
Yep.
And well, from my understanding, they're little short, repetitive, like DNA sequences that you guys used. And like, can you elaborate on that kind of technology you guys used?
Sure. So microsatellites are what we call short tandem repeats in the DNA. So usually it is like AC or AT repeats. So the nucleotides, excuse me, the theory behind it is that as our DNA is being replicated, there's errors in the code. So when the DNA polymerase, let's see, now we're going to go, I'm like 5 years out of this. So you guys got to have to, if it's wrong, or jump in and tell me I'm wrong. So
you're doing all right so far.
Yeah. So basically the DNA polymerase, when it's writing out the new copy of the DNA, when it hits these areas in the DNA that are these repeats that are usually paired repeats, but sometimes they can be triplets or four, but usually tandem. repeats. So AC repeats, AT repeats, whatever, CG repeats, whatever they are. When they're being replicated and rewritten out, the reading and the writing part of the polymerases basically kind of gets stuck on them, and then it starts adding, oh, did I do an AC? Well, I'll add another AC. Not that it has the mind to think through these things, but that kind of process where it's reading, AT, AT, and instead of writing AT, AT, it writes AT, 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 or AT, 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 and continues on. It doubles the entire set. So that's the theory behind microsatellites and short tandem repeats, is this error in the code. So they are, for the most part, what we consider neutral. So they don't have, again, we think this is true, that they don't have any impact on an animal's fitness.
Yeah, no selective pressure.
Yeah, no selective pressure. So it's not providing a benefit or a negative to the animal, the offspring that now has this other mutation. And instead, it's just this kind of, if they occur in neutral parts of our DNA that don't encode for any proteins or anything, then they're just errors that just tend to get replicated again and again and again. So that not only do they get handed on, but then because there's already a repeat there and there's already an error, it has a tendency to create more errors. So these have a tendency to continue to mutate over generations, get longer, but they also can lose repeats. And what this allows us to do basically is these mutations can happen quite, like, compared to mutations in coding parts of our DNA. So parts of our DNA that are encoding for proteins. So whether we can digest, say, lactose or something, instead of it being like that, these things are just happening and they're neutral. So we can see them. They don't have a selective pressure against them, so they don't get knocked out of our DNA. They get handed down from generation to generation. And because it happens a lot more rapidly than we have selective or coding DNA parts, they allow us to see really contemporary changes in a population. And the other is that there's a lot of variations usually amongst them. So depending on the loci, which is that area of the DNA that we're talking about where the repeats are, in that loci, we could have, 2 alleles, simple case. Basically, there's a repeat somewhere. Or we can have, 7, 10, 12 different loci. So they've mutated, lost, gained repeats within that little sequence multiple times. And then you can really tell who's offspring of who, right?
Mm-hmm.
So it just allows us to really look at contemporary. So in the case of my study, the elk were only moved, now 50 years ago, basically. 40 years ago, 50 years ago. So about 40 years ago, those elk were reintroduced to the mainland. And in genetic mutation time, that's not a very long time. But with microsats, we can see those changes within that kind of time period. So they're really good for looking at like new populations, differentiation between, hey, we put a highway in there in 1937. Are these populations still connected? And you'll start to see them separate, because if they're isolated, you can see that in those microsatellite markers.
Recent divergences and changes. Yeah, absolutely are great use of microsats for sure. Yeah, microsats were my introduction into genetics in general, and I don't remember as much as I'd like to on it either. Because at this point, the last time I was using microsats was like 10 years ago for me at this point. And I found it so fascinating that now my career plays around genetics a little bit. So, phylogenetics, but yeah.
Yeah. So now it's kind of turning into old tech. So you're talking about the technology. Well, now it's becoming, fragment analysis, like DNA fragment analysis prior, RLFP or RLFPs, whatever.
Yep. RLFPs. Yep.
Yeah, different older methods that didn't have any sequence information in them. They just showed length difference. And microsatellites are actually kind of like that. What we look at is not the actual, is that an AT? It could be an AT, AC, AT, or some other sequence, but it's the same length. So when we look at microsatellites, it's actually all we're looking at is just the length of the
length of the repeat.
Yeah, the length of the repeat. So it could be different, and that's one of the complications within microsats is that you're taking a best guess that these are two related individuals, and it could be that actually they're not. So there is that odd chance. It's low percentage, but there is a chance that it could be that what we think is two individuals who are tightly related, parent offspring, could be parent totally random animal from somewhere else in the population. So yeah, it's not foolproof. That's what I'm saying.
Yeah.
So now with the most recent technology, which would be like full genome sequencing, that'd be fantastic, but highly expensive for the most part, but getting cheaper all the time. Then we can look and say, oh, well, here's the repeat section I'm looking at. And, oh, you know what? These actually are different. These are not the same animal. So there's a lot more information in next-gen sequencing code stuff. But microsats are still relatively cheap to run and provide really good information for us that we can make assumptions from, and we go with that.
Okay. My next question would be, what would happen in the event of like inconclusive results? Was that something that you guys had to put in the article or the research itself? That what would you guys do with that kind of result? Would you take it out or
like a null result saying that nothing like
yeah that's what she said. Yeah,
kind of. So this particular research and paper we weren't really hypothesis testing, which is a really powerful way, and probably the better way to look at most scientific questions is to have a clear hypothesis and then we test against that. In a way, this genetics work was more of an exploratory. Like, what are we seeing? Now that we know this history, what are we seeing?
Right. Does it reflect the history?
Yeah. Does like what is the result or change that we're seeing, if there is any, from this source population? So it's funny you ask that, because it's one of my pet peeves around research and science is I'm like, we need to have a journal of, can I swear on here a little bit? I won't get too rank, but
a little bit. I mean, eventually we post on YouTube.
Yeah, I wanted to start the journal of shit that didn't work. So
yeah, that's fine.
Basically, research. You guys can beat me out later. So basically, research that had a null result, which I think is as powerful, if not more, in a lot of cases, than actually finding something. And it happens a lot. And then master students or research students scramble to try and find something that they can say did change. And I think it's just it in a way, it kind of undermines the whole process, because it's really hard to publish something that there's nothing there.
Yeah, you're talking about a big issue with that publication bias kind of deal.
Yeah. So anyway, stuff that didn't work. It's a great journal title. And yeah, there was a number of items that we looked at that just didn't show really anything. So we thought that sequential reintroduction. So I don't know, for the people who are online and didn't read the paper, basically, the short history of the project is that way back starting in about 86, they moved a grand total of 27 elk over a number of translocations onto the mainland of British Columbia. From that population has grown. I think now we're somewhere in that 3,500 to 4,000. That's from 27 animals, all told. Wow. So for me, that really, when I was came back to school late in life, and I was sitting in evolution class, and we were talking about population and genetic bottlenecks. And I had worked on this elk translocation or relocation project in 2000 and 2001. I was there right at the start of the second phase, which was reintroduced into the mainland. And then that population grew to about 400 animals, and it was on an isolated peninsula that they couldn't really escape from. So we started having problems, conflict with like the 3 farms that had, vegetable patches on the entire peninsula, but more motor vehicle accidents, elk being in people's yards, ripping out trees, hedges, being aggressive towards people and pets. The population was just getting a little dense for the area they were. There was supposed to be a hunt. As soon as that population hit 80 animals, there was supposed to be a hunt, and they never got it together. There was some conflict over allocation, and so it just got put off. Next thing we know, we have about 400 animals, and of course, they're growing at an extremely high rate. So, few fatalities on the highway. At one point we had an elk with a Zodiac on his head, like a Zodiac inflatable boat stuck on his head, and he was running around the guy's yard trashing his boat. So anyway, all this stuff is like, when we get into wildlife management, these are the things you got to consider outside of just, population, harvest rate, those things. It's also conflict, tolerance of the community, people who live there, other species, things like that. So in 2000, 2001, 2000, we got some money. 2001, we started translocating animals within the mainland from the Seashell Peninsula, this landlocked spot, to other areas around. And there's a whole history in the really brief history in the paper. But if you go and click on my thesis and you want to read ad nauseum about Roosevelt elk history in British Columbia, like a whole chapter just on the history, pretty much. So, So anyways, very successful program numbers wise. Sitting in evolution class one day, I'm like, I wonder what that looks like. And that parlayed then into a master's degree. So, or a master's project. I worked on elk. So can't remember where we were going with that conversation, but anyway.
You were giving us the history on the diversity of the population, which is, I think, fascinating.
Yeah. So we moved them all around. So now there's like 27 elk population units with elk. I think it might be at 28 or 29, 30 even now. A few more population units have since had elk naturally kind of move in. I think the last translocation was 2017. Was the last one that was organized by the biologist that I was involved with, Daryl Reynolds. So, yeah. So and then we were talking about the I can't remember what we were talking about. Sorry.
No worries. I think we were talking about genetic diversity, and you were giving the history of the population and kind of where the idea of the study came from, which sounds pretty basic because it sounds pretty good because you had 27 elk that are now responsible for most of the genetic diversity of, would you just say 29 populations?
Yeah.
That are so 27 elk from various locations all put onto one island, and now they're responsible for 28, 29 populations of elk.
Yeah. So the mainland is an island.
Right.
They came from an island.
They came from the island. Yeah. That's what I meant. Yeah. Context is important, right?
Yeah. So we were talking about microsatellites, and that's why I think I went on this sort of tangent on the history. So when we moved them from this peninsula and another population that had been established a little bit later in 96, we moved them from conflict zones to isolated areas. So we moved them in winter, which is when we could bait them easily, but it's also when in the coast of BC where these elk are is like fjord land. So it's very deep, long inlets that go from basically temperate tidewater all the way to ice fields, like right below ice fields, a number of them. So the elk, when we dropped them off in winter, really couldn't walk back. And surprisingly, we documented a number of times when we would drop some off and then go back to survey within a couple of weeks, they would try and beeline, like you hear about bears just going straight back to where they were dropped off. Those elk wanted to do that exact thing. We'd drop them off and they would turn and go direct line back towards the peninsula from wherever they were. Most of the time, they got stuck in deep snow, impassable cliffs, other things that make it very hard in the wintertime to get out of some of those inlets and valleys. In the summertime, most of them have low passes at the back that they can get out and get into the neighboring valleys, or they can walk along the coastline and avoid falling in the water from snow and stuff. So one of the other questions within that, and where microsats are really good, is you can look at very contemporary isolation. So we were also wondering, In the 17 years we translocated elk throughout the mainland, how connected were these little populations we tried to establish? So the reintroduction method used was basically try and get at least 20 animals into each new spot. And that was from a paper that talking about reintroduction strategies. I can't remember the name of it off the top of my head. It's in there. That the government used as, okay, well, it says we need for genetic diversity at the time, which was our understanding was a lot more limited than it is today. We thought 20 animals, that gives us a really good chance of catching most of the genetic diversity in a population to introduce somewhere. Okay. So that was kind of the threshold. We did everything we could, and the biologists, after I left that program in 2004, they continued on trying to get at least 20 animals into every population. Now, the theory behind that is 20 effective breeders is really the idea here. You have an effective population. So have you guys talked about like those genetic measures, diversity measures much in your class?
We don't focus on most population genetics in genetics, so defining terms is probably helpful for Hayley and Ruby here, even though I think we've talked about it in other classes, but I'm not sure if they were part of those classes or not.
Well, and for the audience too, who don't.
Yeah,
maybe don't.
exactly.
So when I say effective population size, when we talk about Roosevelt elk, we may have 1,000 elk, but the effective population size might be 6, 12. Basically, what it means is we have the genetic contribution of what we would consider 12 unrelated individuals, for a really rough kind of way to put it. Mm-hmm. So in the case of these reintroduced populations and most populations that have undergone some sort of significant bottleneck, everybody's cousins of cousins of cousins. So everyone's related. And what that does is it means that the effective number of individuals just starts to dive. And I can't remember if I calculated out an effective population size. I think I did. Anyways, for most of those populations, but most of them were tiny, like 2 individuals, 4 individuals, 1 individual. Roosevelt elk are already, because of historic population bottlenecks, they already have very low diversity. And that causes some problems when you try and do some other analysis around bottlenecks and things. Because it kind of, all the genetic measures really look at the population you have on hand. They don't really compare to the population you used to have before the bottleneck. There's only been a few papers that have been able to do that. One I quote in there is a study on sea otters, if anybody's interested in reintroductions and stuff, and what genetic diversity looks like in populations that have experienced significant bottlenecks. That's a great one. Can't remember the author off the top of my head, but there's sea otters on the west coast. If you just look up genetic sea otters, it'll probably be the first or second paper to pop up. And one of the papers, so they did one kind of like mine. They had done a paper where they just looked at mitochondrial microsatellite DNA amongst all the surviving, or all the contemporary populations of sea otters, and sort of said, we see this diversity, and we can see that, these animals, this one population has animals that came from California and animals that came from Alaska, and we can see that there's this really high diversity in this population. But then, so it looks not bad when you look at all the diversity metrics. It's like, that's not bad. Your heterozygosity is up over 0.5, so it's 0.7, 0.8, whatever. Then they went back a few years later and they got a whole bunch of archaeological samples from middens up and down the coast and looked at bones and things and extracted DNA from pre-bottleneck, so pre-fur trade or early fur trade. And then they compared, and it's like night and day. There's like nothing left. There's no diversity left. It looks great when you only look at the one population, but if you look at what used to be there, it's horrifically low. So it's an interesting couple papers. Larson, I think, 80s, 90s. Anyways, and some work's been done on Rocky Mountain elk, or elk in general. Wapiti in general in North America that kind of shows the same thing. So we had, and some people still do, we have Canadian elk, Rocky Mountain elk, Coulee elk, Roosevelt elk. What's the other one? There's a couple others. Eastern elk. There was a number of what we thought were subspecies of elk had originally been designated subspecies. And and Polzine and a number of other authors have looked at elk genetics like continent-wide. The only ones that really hold as subspecies is, well, we have Rocky Mountain elk or Canadian elk, whatever you want to call them. Coulee elk in California and Roosevelt elk. So those are the three that still had sort of supporting documentation or some supporting genetics to say these are subspecies. Roosevelt elk and Coulee elk are subspecies of Rocky Mountain elk. And you can get into this whole other thing about subspecies.
Yeah, that's what was going through my head, but I was gonna save you from my usual comments on the subspecies debate.
Yeah. So from a phylogenetic point, is Roosevelt elk and Coulee elk are isolated and their own genetic groupings in most places. If you stick a Rocky Mountain elk in there, you're going to have some interbreeding, that's for sure. Now, whether they're adaptive, that's what we really talk about when we talk about subspecies, I think, or we should anyways, talk about the adaptive potential or the how adapted they are to their specific environment.
Oh, yes. I see what you're saying. Yeah.
And then say, yeah, this is actually a subspecies. Like, so we looked at, I think if we took a Roosevelt elk and we dumped it off in Western Alberta, Rocky Mountains, or we took one from Western Alberta and we dumped it off on the coast, both of them would be dead pretty damn quick, is my opinion. Because just because I think they're genetically different enough and have probably adapted enough to their environments to thrive where they are, and probably not thrive if you move them around. So, yeah, Roosevelt elk, I don't know if you guys are probably not super familiar with the West Coast, but if you go Washington State north, it is wet, it is cold, it is awful. The soils have no nutrition. How Roosevelt elk get as big as they do living on the vegetation that's available to them it's crazy, really. But it must involve a lot of small adaptations to being able to digest and thrive off of sword ferns. Like, I see them eating sword ferns, and sword ferns have,
it's like a nothing. Yeah.
No nutrition. Yeah, they eat tips off them at a certain time of year for a couple of weeks, and it's in that like starvation season in the spring, and they seem to do okay with it. So, yeah, it's the only animal that's probably more adapted to eating everything and anything is the black-tailed deer that live in the exact same ecosystem. They eat everything. So, yeah. So that's where I can look at subspecies that way. And then we get into birds.
No,
Who even knows?
No.
All right. My next question would be, what do you hope other readers would notice on the article?
Other than what you guys noticed?
Yes.
I mean, you can agree with her too, and we're not going to fault you for that.
Yeah. No, I think you guys did a good job of grabbing that, sort of like narrowing it real down to its essence of what's there, which is that there is the potential for a problem here that we should probably keep an eye on. What's really the sort of the
crux,
end, the end note was, even though, like, so it's what I believe is happening with elk and Roosevelt elk specifically, and is kind of not missing, what's the word I should say here? Underexplored avenue in conservation genetics of small populations and at-risk populations is this capacity for what we call shedding deleterious alleles or cleansing of the genome for deleterious alleles. So if you guys are doing genetics, you understand the concept that.
Yep, we've talked about deleterious alleles a bit.
Yeah. So basically, alleles, like changes in the DNA that have even small effects on an individual's fitness, as they get passed down, that then can have large effects on a population's ability to continue and thrive and reproduce and grow. So if I think probably one of the most famous examples is Florida panthers, where, very small population suppressed down to a very small population for a very long time. And pretty soon they figured out that even though they have some males, turns out some of those males were sterile. And these, from mutations in their genome, well, you're just not getting any young reproduced. So they ended up reintroducing, if I remember correctly, from like Texas or somewhere.
It was Texas.
It was basically the closest, genetically closest group to this group of Florida panthers. They brought in some males, let them go, boom. Suddenly you get this kind of like, almost like hybrid vigor, but it's not hybrid vigor, but it's reintroduction of healthy genes into that pool that kind of then are able to swamp out some of that deleterious alleles. So if you get, the problem with small populations, you've got a deleterious allele, maybe you can't digest something very well. And then the mate you run into also is carrying that deleterious allele. And if you only got one allele each, right, like you could have one or another, you're both carrying the same one, well, all your offspring are having that same one, if that's the one you pass on. Or not all, but, a quarter at least are going to have a double homozygous at that location. And then that's a problem. Now you've got individuals who are not thriving. In elk, and these elk kind of really show that, is that, we've got these bottlenecks, huge bottlenecks, and this really reduced genetic diversity. But damn, they're growing like crazy, and they are the biggest things you've ever seen. So I could pull up a picture, but we saw an elk a few years ago flying goat surveys. That I swear was 1,500 pounds. Like, he looked like a Hereford bull. He had no neck. And if you know anything about elk, is they have this big long neck that everybody shoots them wrong when they're hunting them. They shoot them in the shoulder. They think they're behind the shoulder, but they got this big long neck that makes them look like front heavy. So, and this thing looks like, his neck looked like this. Like, it was like a Hereford bull. He had no neck at all. And I know how long his neck is. His neck's like 3.5, 4 feet long, and you can't see it. It's just, I swear, if you poked him with a pin, he would have popped. His skin would have flown off. He was so fat. But anyway, so these are like the largest elk ever weighed was like, I think, 1,240 pounds or something up in a Fognac Island in Alaska. And we weighed one on the very first year, some of the first bulls we captured in March. So that was in hunting season. So September on a Fognac, 1,100 or 1,200 and something pounds. And then we weighed a bull that was starving to death, looked like an old nag, his hip sticking out, every rib sticking out. And he was 1,180 pounds without his antlers. And he looked like skin and bones. So it would not surprise me to see a bull out there over 1,500 pounds, like the size of a large moose, a good moose. So, yeah, they're huge on the coast. And again, I don't know what they're getting nutrition from, but they're digesting something well. So,
I have a question on that as well. Like, since the animals were so big, how would you guys relocate them?
So we had, so when we started, so jumping way back, so 86 reintroduction, dropped off Seashell Peninsula. That population, they dropped them off in a logging slash, which the next year they developed into a golf course. Perfect for the elk. They love that. They love golf courses. It's open. There's no brush. They can stay dry. They're not constantly. So, and it's a great place to fight and set up dominant contests. Yeah, kind of like a lek. So the Pender Harbor Golf Course became ground zero for all Roosevelt elk activity on the coast for a lot of years. And to the point that they put in like a 12-foot fence around the whole thing because they got tired of reseeding everything every day because the rut was off. So because that population was so problematic where they dropped them off, it was the first one where they thought, you know what, we just got to move some of these elk. Like, this is crazy how many are here. So on the golf course, they built a big wooden trap, a corral, about 10 feet high out of like rough cut 2x6s and stuff. It was weighed an inch. It was huge. Anyways, we that was used in 1996 to move, I think, 11 or 12 animals from the peninsula to a place called Paul River. That was a new reintroduction. Then they brought another, I think that's when they brought 5 more from Vancouver Island in 96 to Paul River. So they used that trap, I think, once or twice, and then it just sat. So we reclaimed that in 2001, and we set it up nearby where it was originally set up. And with a whole great group of volunteers, the Sunshine Coast Rod and Gun Club provided, I think we had 30 or 40 volunteers come out, help us rebuild it, reset it up. And we got some of the money we had acquired from Rocky Mountain Elk Foundation, which used to have a presence in Canada up until about 2008 or 2010 or something, I think. They gave us some money we got an old truck, an old, what do you call it? Like an old transport truck. Or we hired a few guys from different places too that had like cattle trucks and things, trailers. But anyways, we had set that up, and we baited it with apples and corn sugar and molasses and alfalfa and you name it. We stuffed it in there to see what would work. And I think the first capture we got 15 elk or something, which is way too many. So you don't want to go. And put them on a barge and took them to the opposite end of the Sunshine Coast. And, so we used that trap a few times. We built a new one and set it up on one of the golf courses in the neighboring town of Seashell. That's where we caught mostly bulls. And then we were like, man, we got to get these traps they're just not mobile. So we got some funding again from Rocky Mountain Elk Foundation and a bit from, I think, Habitat Conservation Trust Fund in BC to build chain link panels. So we had all these chain link panels built and ramps and gates and things. And those were good. We could stack them. I think they were 7 feet wide, so we could put them on a flat deck trailer, and 10 feet high. So we could stack the entire trap on one trailer with gates and everything, and take about 4 people to set it up. So it reduced our required workload a lot. And set up a trigger, which we used to have set like a remote. You'd set a string across the back with a solenoid to prick the trap when we hoped there was animals in there. And then like the last year I was on that project, I got hired as the coordinator for government to do the baiting and all that stuff. And we were talking about that. Like we had some groups that were 7, 11 animals, something, and they were real problematic. We didn't want them still there. We wanted them out of the neighborhood or away from the place. So I started climbing up a tree stand and then running a string to the release, and just sitting there freezing to death in the rain for hours on end until the elk showed up to walk in and make sure we got them all in one go. So we did that quite a few times as well. So yeah, the trapping is interesting, sometimes a bit of a rodeo, but yeah, there's nothing like getting it in the back of a truck with 12 not very impressed elk that weigh 700 pounds each and their offspring, and cows will grind their teeth. This like sound, they'll just grind their teeth when they're getting really mad, and you can just hear it around you. They're all really mad. There's lots of them making that noise. So yeah, it was some fun work. It's back kind of before we really got into the whole occupational health and safety kind of stuff for the government. They don't really like you doing that now. We have like these little 1/4-inch plywood shields that we built to try and do it, but we're putting collars on around the neck of a untranquilized elk in the back of a truck with 10 or 12 or 6 others, and that was some interesting work. So for sure.
Wow. Can you elaborate on the impacts you and your team hoped it would have? Do you feel that this was accomplished?
Yeah. So Daryl Reynolds was a biologist that really spearheaded the whole, not the original translocations to the main, like the original reintroduction to the mainland, but all the translocations from then on within the Sunshine Coast. And that was, again, it was to alleviate some of the conflict and pressure. And also because the original purpose of the reintroduction to the mainland was because the Roosevelt elk on the island are so genetically, like they knew they had some real basic genetics on them. But from that, they looked like, I think it was all RLFP stuff, fragment analysis. They were like, these are all one individual. Like, we have no diversity. So the original stuff, they couldn't tell individuals apart at all. The original work that was done. So they were really concerned that should any disease break out, so somebody brings sheep or alpacas or something to Vancouver Island, and the disease switch of species and jumps into the elk population, the potential is to lose everybody because there's not enough, maybe nobody has enough diversity to survive. So that was the real concern back in the late 70s, early 80s. They were like, man, this population is at high risk of extirpation and loss. So they set up that original Sunshine Coast population really as a reservoir. Should something horrific happen on the island, they could reintroduce elk from the mainland back to the island. So it was an isolated population that would be able to escape any disease issues that may come up on this connected populations on Vancouver Island. So that was the original goal. And then we got this conflict goal of issue of conflict, and we have historical records from First Nations and early explorers of elk kind of up and down the mainland part of the coast. Anywhere there was a big river valley, so the Squamish River, the Fraser Delta had probably thousands of elk running around on it. Pitt River, all the rivers that kind of flow into Georgia Strait and by Vancouver Island, but also some of the bigger rivers all the way up to like almost to Bella Coola, I think. There was reports from early explorers written down saying there's elk here. And the assumption was these are Roosevelt elk. So Darryl was really like, we've got two things happening. One, we got conflict. We've got a suppressed population that's stuck on this little spot. And we've got all this habitat that has been created by intense forestry. So the entire west coast of BC, like the west coast of everywhere, anywhere there was trees, they cut most of them down. So lots of big slashes, river, valley bottom openings. And we just saw it as this huge opportunity to increase that population beyond trying to keep it at 400 animals. Are we limiting ourselves to 400 animals? Let's create opportunities for First Nation harvest, recreational hunter harvest, ensure that the population has lots of animals to choose from should a disease outbreak happen somewhere. There's lots of elk to capture here and there. Turns out they also like silviculture, so replanting trees. When you have like low nutrient forage everywhere, when they plant trees on the coast, they put a little fertilizer packet because the soils are so bad, and then those little trees have great nutrition. So they would go around and pull them all out of the ground or nip the tops off. So we ended up creating some conflict with forestry. Some of them were not very happy with this. Yeah. So it was a couple, and then there was the whole thing about were the elk ever there, and I think it's pretty clear from the historical record that yes, there was elk on a lot of the mainland coast, anyways. So we managed to get them back into most of their sort of historic distribution, and I think that was it's just been a resounding success that way. I think I talked to Darryl, but I think there are over 100 or 200 tags, maybe 200 tags now on the mainland. For recreational hunters, and then there's almost an equivalent amount available to Indigenous hunters or First Nations harvest as well. So for a lot of those bands, that was an important thing for them. They wanted to establish that tradition of hunting elk without having to travel all the way to eastern British Columbia or something in their own territories.
So I want to pivot us just a little bit. So what is your favorite way to communicate research findings? You're so well spoken in this so far. You're going into some amazing stories, and I'm loving it. But I want to know, has this been enjoyable so far, or what's been your favorite way to ever convey results of work you've been doing?
Yeah, so the elk work I've presented a few times, wildlife society meetings and stuff like that. Just on a really kind of, like this sort of, very non-technical, not like presenting a paper and just talking about the issues and stuff. And I like that. I like that very casual communication. I used to hate public speaking completely. And now I'm like, I'll talk. Sure, man, I love talking about this stuff. So I always tell anybody, like, okay, you can ask me about elk if you want, but I hope you're ready for to sit down because we're going to have a conversation. So Yeah, I like talking about health and sheep and all these things. So, so I did a fair bit of public speaking, presenting to fish and game clubs or, naturalist society group meetings, or any way to get information out there. I think is always beneficial. Get your message out. That I was kind of working on a website or Facebook page for my other work on bighorn sheep that I was doing. But it's just, man, it just takes time. So, I got great intentions at the start of some of that communication. In the end, not so great. So I really appreciate people who can write like plain language articles for public consumption in Outdoor Life or, on a blog post or something, just talking about science, but putting it in terms that people can really understand the message without getting lost in the techniques and technological and the numbers, which is easy to do when you're immersed in it for years on end, trying to figure stuff out. A lot of us get stuck talking about sampling methods.
No, we don't need to get caught into those weeds for sure. No, there are methods papers for a reason. We don't need to go dig into those papers. But I think we're at a point where we can start with the wrap-up questions. So if you guys would like to ask the last three questions.
Yes. I would like to ask, what would be one piece of advice you would give your undergrad self?
What? I think from my transition from undergrad to master's student was pretty seamless. I did an independent study, genetics independent study in the lab that I ended up doing my master's in. And actually I'll give everyone a piece of advice that I got, which helped hold me through that. I was talking to a fellow that I knew, real smart, like smart as a whip guy. I run into him at the university one day and I'm like, hey, what are you doing here? Like, I hadn't seen him since college. And he's like, oh, I just finishing a master's. I was like, oh, I didn't know you did a master's, man. Great. What did you do it on? So it was like on parasites and fish or some damn thing. Anyways, I was like, oh, well, I'm thinking about doing a master's. Like, any advice for me? And he's like, yeah. Only do a master's if you are just enthralled with the subject matter. Like, you've got to know it yourself. Like, you got to have that self-interest. And I was like, oh, okay. He's like, I just did one on something. I did it because it was an opportunity to do a master's and I thought I needed one to get a better job. So I did it. He's like, it's been 2 years of hell because I don't really care at all about the subjects I'm working on. He's like, and it's been just a nightmare trying to finish because I really don't care. And I was like, oh, okay, that's good advice.
That's the advice we've gotten previously is follow your passion.
Yeah. Follow your passion. So the lab I did my work in is a bird lab. They study birds, genetics of birds, almost nothing but a little bit on plants and other ancillary kind of things surrounding. But I walked in with Prof and I said that I got two ideas. One is sage grouse genetics on our sage grouse in Alberta are critically endangered. There's like hardly any left, but they were doing a big reintroduction project there where they were bringing in animals from northern Montana. And I was like, we don't even know if we have any Alberta genetics left in this population or not. So that might be an interesting one. And there was funding for that, like lots of funding. I was like, but there's this project I used to work on like 15 years ago that was really got me into going back to school. And I would really like to answer this question about what happened with this reintroduction. What does the genetics look like? And thank God I picked that one because I was really motivated because I had a history with the project to find out the answer to it. And that's what kept me going because a master's is a slog. I don't care who's talking about it. If you aren't like ready to quit at least a dozen times, you ain't doing it right. So it's a lot of work. It's just all consuming. I don't know if there's an easy way through it. There probably is, but I didn't find it. Nobody I know has ever found it. Everybody I know who's done a master's has just been, man, it's a slog to get through. And you're done, you're super happy, and you get some great work on just stuff out of it. But the getting there is a stretch. So if you're gonna continue on after undergraduate, make sure that whatever you do, you're gonna follow up or continue to work on is something that you're passionate about, that you really want. Like, just love. Like, that's why there's so many people work on birds. So many people just love birds, right? So yeah, there's lots of bird work going on all the time.
Birds and fish.
Yeah. Yeah. Well, and there's, fisheries work on. You just got to catch things and samples. Yeah. Yeah. Unless you want to catch like marlin or giant bluefin tuna, in which case it's kind of like working on elk. It's 800 pounds and you got to handle this thing. So yeah, that would be that one advice. And then, sorry, another question.
Yep.
Oh, yes. So what's one thing you wish more people understood about science or scientists?
Oh, that's a good question. Well, I'm going to go on a tangent. You're probably not. I won't put a tangent. I'll go on a tangent, an angry tangent. Yeah. So one of the things that I'm sure you guys are well aware, there's lots of discussion around settled science on a bunch of shit. No, science doesn't settle on anything. So we talk about hypotheses and theories. There's theories that are quite well proven and supported. There's still always the question, maybe that's not what's happening. So, the best theories we have are with what we know at the time, with the tools we have at the time. And those are always changing. So every time I hear somebody say the science is subtle, I just want to punch them in the nose. Because, yeah, science is just about curiosity and the search for truth, reality, whatever you want to call it. It's about trying to find out what's really going on, not proving anything else. But unfortunately, financially, like I was saying about the sage growth thing, I got no money from my elk research. Thank God my supervisor, Teresa Berg, she just helped me out by giving me just enough to basically cover all that. I had lots of scholarship money for myself, but it didn't really give me any lab supplies or anything. So she was able to help me out with lab supplies and all those kind of lab costs. Otherwise, it would have fallen apart because I would never have got the money. Sage grouse were highly endangered in Alberta, and there was money left hand over fist for that, but I wasn't as interested. So that's where you kind of got to weigh it out. It's an interesting question, but not as interesting as elk. So yeah, if you can, follow your passion.
Absolutely. So I think we'll kind of call it right there. I want to thank you, Ian, for taking the time and talking to us. It's been a real pleasure. I'm glad we were able to make this work. For a while, I was like, oh, it's not going to happen. And I thought we'd have some good stuff to talk about. I'm sorry we didn't actually. I was hoping we'd dabble into some of the bighorn stuff because we do a big bighorn thing out here. Yeah. But in our populations we have down here, which would have been interesting to get into, but we just don't have the time at the moment. But I thought that would be fun. Yeah. But thank you so much.
I wasn't doing any genetics with cheap.
Well, that was it. we could have made something work. But I want to thank you so much for taking the time. I want to thank Ruby and Hayley for taking the time and putting together the questions to get us going on things and finding and going over the paper. And with that, this has been STEMpaths, and we'll see you guys next time.
Thank you for the opportunity to talk, and it was very good questions. I appreciate them.